The last group of invertebrates that we need to cover are the deuterostomes. Deuterostomes are characterized by their development of the blastopore into the anus and then the other end as that blastopore extends into the mouth. Deuterostomes are important because this group is going to lead us to our chordates, which we're going to explore a bit more when we get to chapter 27 and our vertebrates. Our deuterostomes can be broken down into two phyla, the echinoderms and the chordates. The echinoderms include our sea stars, sea urchins, and sea cucumbers. And these guys are a little bit interesting because they appear to have lost some of our previous characteristics that we see in our protostome groups. And then if we look a little bit closer, though, we see that they're actually there. They're just in some different forms. When we get to our chordata group, our chordates, we're going to talk about some very general characteristics for the chordates, and then we'll jump into the vertebrates in chapter 27. So starting with the echinodermata, we're going to sort of use sea stars as our prime example of phylum echinoderm. A sea star is basically 5 points, like a star, and it would appear that they are not bilateral or radially symmetric. It turns out, if you follow the lifestyle, that the larvae are bilateral, which is why we put them into the bilaterians. The radial symmetry or modified radial symmetry that comes up is a secondary feature. So as those larvae develop, they switch from bilateral larvae to radial structure. They have a modified radial symmetry, so they have 5 points. 5 points is not radial, so if we have a sea star, we cannot cut this on any plane and get the exact same thing. We might be able to get some bilateral symmetry depending on how we cut it, but we don't really have a true radial symmetry because we have the 5 points. So it's not a true radial symmetry. It's not really a true bilateral either because there are multiple planes that we can cut and make different bilateral planes throughout the organism. So it's kind of a weird structure, but we still consider it bilateral because of the larvae. They have no brain. They have no cephalization, which is different than everybody else that we've talked about in the bilaterians so far, the Lophotrochozoa and the Ecdysozoa. They all have what we would consider a head end. So these guys appear to have lost that as well. They have a very simple nervous system. They still have the tissues, they just don't have some of the structures that the other ones do in this group. Echinoderms have some unique features that we haven't really seen yet in our organisms. One is an endoskeleton. So endoskeleton basically means inside support structure with an outside covering. That outside covering is often covered with spines and structures called pedicellariae. So spines are obviously sharp. They're to deter predators and for protection. Pedicellariae are basically little pinchers that occur on the outside of the endoskeleton. So there's an example here of a spine in the center with pedicellariae on the outside. So you can see those pincher-like structures. So that's going to be for protection. Usually echinoderms have a lot of these protective features because they don't move very much and they need a way to defend themselves against predators. They don't have any excretory organs. Their respiration and excretion or waste removal is done by diffusion. They can also detach a body part and regenerate. So kind of cool. Lastly, at least what we're going to talk about, they use something called a water vascular system for movement. So basically it is this tube structure inside the organism that spreads throughout out to the arms or whatever structure they happen to be, whatever they're using for mobility. And it produces these little tubes that come out of the endoskeleton, and using water pressure and changes in water pressure, it causes the tubes to move. So they use this to move slowly around their environment. If you've ever picked up a sea star, on the bottom you might see those little like tentacle-like structures. Those are actually the tube feet that are moving by this water vascular change of pressure mechanism. Here are a couple of examples of echinoderms. If you've ever been to a touch tank at a zoo or an aquarium, you've probably seen some different examples of echinoderms. Some of them get really thick. These examples here, the necklace star and the brittle star, are real thin. We've also got a sea urchin, and then a couple others, a sea lily, which is really kind of cool, and a sea cucumber. Just like some of the other vertebrates we've talked about, there's some really cool examples if you want to look them up. If you look at the necklace star, you can see these sort of white structures on the organism. That is most likely going to be where their spines and pedicellariae are located. You can't see that on all of these organisms, but it's a very common feature of echinoderms. Lastly, we're going to talk about our chordates. Our chordates include all of our vertebrates and some invertebrates. All of the remaining organisms that we're going to focus on in this class are going to be our vertebrates, and we're going to focus on those in chapter 27. But chordates are unique. They have some really specific characteristics that lead to the development of what is going to be a spinal cord and a vertebral column eventually as we travel through our different types of organisms. So common distinguishing features of the chordates are the notochord. We'll talk about that in just a second. The dorsal hollow nerve cord, pharyngeal slits, and postanal tails. And one thing I would like you to keep in mind is that these are going to be present in all of our chordates, but they can be present at any life stage. So for example, during development, humans have a postanal tail, but it is basically sort of gone by the time we get to birth and then development. One of the bones is still there, your tailbone or coccyx is still there as sort of a remnant of this postanal tail. We just don't see it in the adult form, but we do see it in the embryonic form. So as long as it has these at some point in their development, they count as a chordate. So our key example of this is called a lancelet. We also see the same characteristics in a tunicate. In our lab, we looked at Amphioxus. We have several different things you can look at if you want a reminder. We've got a model, we've got slides, we've got a resin preserved organism. The Amphioxus, or the lancelets, are our prime example because we see all of the early versions of these different characteristics. So we have the notochord, which is this cord here. In our later chordates, the notochord is going to become the vertebral column. In other words, all the bones that surround your spinal cord. The dorsal hollow nerve cord is going to become your spinal cord, or it's going to develop later into the spinal cord. And also the brain. So right now with the tunicates, Tunicates and lancelets. They don't have a very well developed brain in the sense that it is not as large as we're going to see in many of our other animals, but they do have a centralized location that we would consider a brain. So that dorsal hollow nerve cord is going to develop into the spinal cord as we move through our organisms. And then, of course, we're going to have a more extensive brain development as we move along. We also have pharyngeal slits. Depending on the type of organism, the pharyngeal slits are going to develop either into gills in our organisms that have gills, so like our sharks and our fish, or as we get to terrestrial animals, in our terrestrial animals, they are going to develop into some parts of the head and neck, specifically the ears and part of the jaw. So the development of these structures are going to be just a little bit different depending on what organism we're developing into, but we're going to see those pharyngeal slits as well. Again, we don't have pharyngeal slits during embryonic development. The embryos do show pharyngeal slits, but they eventually develop into the ears, the inner ear, and the jaws. And then lastly, the postanal tail. You can see that here. Postanal just means at the end of the organism, behind the anus. So the region behind. You guys are all familiar with this in many, many animals. Our lizards have them, cats have them, dogs have them, etc. Humans don't have a postanal tail after our embryonic development, but again, it's present at some point. The lancelets and tunicates are filter feeders, meaning that they siphon water into their mouth structure, or through the top, acting as a mouth at the top, and then absorb their nutrients on the inside.